Good morning, everyone. My name is Michelle Parker. I'm a technical writer, marketing specialist, and antibody services at GenScript. Thank you all for attending this webinar on anti-idiotypic antibodies and their use as powerful tools for antibody drug development. To ensure the sound quality, all of you are muted to minimize the background noise. If you have any questions during my talk, please send your questions through the chat box at the right lower corner of the screen. I will address your questions all together at the end. Here's a little bit about my background. Like most of you, I came from an academic research background. I obtained my PhD in pharmacology and toxicology from the University of the Sciences in Philadelphia. My dissertation focused on the design, engineering, and testing of a recombinant albumin fusion protein drug delivery technology. Prior to graduate school, I spent a number of years studying the cellular aspects of cholesterol metabolism at the University of Pennsylvania and Children's Hospital of Philadelphia. It is my hope that this webinar will give you useful information regarding the different aspects of successful anti-idiotypic antibody development, as well as the use of anti-idiotype antibodies in complex assays for antibody drug development. This presentation will begin with a brief review of antibody structure and function that will segue into the description of what constitutes an anti-idiotypic antibody and the variety of applications in which they can be used. If you are at all familiar with anti-idiotype antibodies, you will know that their successful development comes with a number of obstacles. We will dissect these issues and provide solutions which have allowed our scientists here at GenScript to have a 100% success rate in anti-idiotypic antibody generation. The logical next step after successfully producing high specificity, high affinity anti-idiotype antibodies is the design of drug monitoring assays for human or humanized monoclonal antibody drugs. There are a number of methods for detection, quantitation, and characterization of antibody responses, which we will review in detail to help you advance to the next stage of your project. In the end, I will go through some of our customers' most valued antibody services and how GenScript stands out from our competitors. In order to set the stage for our discussion of anti-idiotype antibodies, we will first briefly review the structure and function of antibodies in general. Antibodies come in millions of different amino acid sequences, making them inarguably the most diverse proteins known. Each antibody consists of 4 polypeptides, 2 heavy chains and 2 light chains, which are joined to form the Y-shaped molecule. The amino acid sequence in the tips of the Y vary greatly among different antibodies. This variable region, composed of 110 to 130 amino acids, gives the antibody its specificity for binding antigen, while the constant region determines the mechanism used to destroy antigen. It also forms the basis for the division of antibodies into 5 major classes: IgG, IgA, IgM, IgD, and IgE. You may also note from the illustration here that the variable region is further subdivided into hypervariable and framework regions. The hypervariable or HV regions directly contact a portion of the antigen surface. For this reason, they are also sometimes referred to as complementarity determining regions, or CDRs. The framework regions serve as a scaffold to hold the HV regions in position to contact antigen. Here we will dive further into the antibody binding regions to lay the foundation for our discussion of anti-idiotype antibodies. The idiotope of an antibody is the unique set of antigenic determinants, or epitopes, of the variable region of an antibody. It can be the actual antigen binding site, and in some cases it may comprise sequences within the variable region that lie outside of the antigen binding site on the antibody itself. Thus, it is possible for one antibody to have multiple idiotopes. The paratope is the part of an antibody that recognizes an antigen, the antigen binding site of an antibody. It is typically a small region consisting of 15 to 22 amino acids located in the antibody's FV region. The paratope may also contain parts of the antibody's heavy and light chains. The epitope, which is also known as the antigenic determinant, is the part of an antigen that is recognized by the immune system. It is the specific part of the antigen that an antibody binds to. It is the set of individual idiotopes that is termed the idiotype of an antibody. An anti-idiotypic antibody is an antibody which has specific binding capabilities to an idiotope of another antibody. As mentioned in a previous slide, there are three hypervariable regions coincident with antigen binding sites. They are referred to as the complementarity determining regions, or CDR. Since the anti-idiotypic antibodies target the CDR's antigen binding sites, the anti-idiotypic antibodies may or may not compete with the antigen. With these characteristics, anti-idiotypic antibodies can be powerful reagents for a number of applications. An important aspect of anti-idiotypic antibodies is the ability to perform guided selection during the development process to isolate anti-idiotype antibodies with rare specificities. For example, drug-target complex binders depicted on the far left can be used to quantify bound drug as opposed to free drug levels. These complex specific antibodies bind neither the antibody drug nor the drug target and can therefore be used to detect bound therapeutic antibodies directly. Likewise, neutralizing or antigen-blocking anti-idiotype antibodies, illustrated in the middle, as well as anti-idiotypic antibodies that bind to an idiotope outside of the antigen binding site, labeled as non-blocking on the right, can be selected. We will see later how specialized binding modes of anti-idiotype antibodies can aid in assay development. We have discussed the characteristics of anti-idiotype antibodies and their diversity, which offers assay developers enhanced flexibility in assay design. But why are anti-idiotype antibodies such powerful tools in the realm of antibody-drug development? The answer to this question begins first with the consideration that most biopharmaceutical therapeutics elicit some level of antibody response against the product. This response, in some cases, can lead to potentially serious side effects and/or loss of efficacy. The process of monitoring therapeutic antibodies in samples, however, requires the ability to differentiate between administered antibody and naturally occurring endogenous antibodies. This process has become increasingly difficult as antibody biotherapeutics more closely resemble circulating human immunoglobulins. Anti-idiotype antibodies, specific for the unique variable region of the therapeutic antibody, are ideal for highly sensitive detection purposes. Anti-idiotypic antibodies can be used in a number of different capacities for drug development studies, given their unique ability to identify drug antibodies in biological fluid. When a human or humanized antibody drug is administered into a patient, the drug becomes hidden within the high concentration of very similar antibodies in human serum. Typically, the concentration of the antibody drug can be 1,000,000 times lower than the serum antibody concentration, making the drug difficult to detect and quantify. Consequently, preclinical and clinical development assays for these antibody drugs require highly specific as well as highly sensitive detection reagents to measure their pharmacokinetics and immune responses in human subjects. Anti-idiotype antibodies are indispensable reagents for pharmacokinetic studies to quantify drug level in patient samples and immunogenicity assays where they are used as a positive control or reference standard. In addition to being powerful reagents for drug development studies, anti-idiotypic antibodies have also proved highly useful for vaccine development. Anti-idiotype antibodies can be classified into two main groups, AB2 alpha and AB2 beta. Amongst them, the AB2 beta antibodies, which constitute about only 15 to 20% of the total anti-idiotype antibody, carry an internal image of the nominal antigen. The internal image of antigen-competing anti-idiotype antibodies can mimic antigens and thus may serve as immunogens for vaccine development, especially if the antigens are toxic or infectious. Before we discuss the obstacles to anti-idiotype antibody generation, I will briefly review the process involved in monoclonal and polyclonal antibody development. Let's first take a look at monoclonal antibody production. Monoclonal antibodies are made by immunizing a host animal, usually mouse or rat, with an antigen, and then fusing that animal's spleen cells with myeloma cells to create immortal hybridoma cell lines. The cells are then grown in a selection medium called hat medium. Only fused cells, hybridomas, can survive in such selection medium. The most productive and stable hybridoma clones can be screened out for antibody production, and purified monoclonal antibodies can be obtained from the supernatant of that hybridoma culture. Let's now take a look at how polyclonal antibodies are made. Polyclonal antibodies are generated by first immunizing host animals with antigen. The host animals can be rabbit, sheep, rat, hamster, horse, etc., depending on the amount of polyclonal antibody needed, as well as the closeness of species between antigen and host animal. After the antigen is injected into the host animal, the antigen is then digested by the macrophage, where it can then be presented and recognized by B cells and helper T cells. The B cells replicate to produce plasma cells, which are the antibody-producing cells. Serum containing these antibodies is then collected from the host animal, and purified polyclonal antibodies can be obtained by immunoaffinity purification from serum. One can generate both monoclonal and polyclonal anti-idiotype antibodies. However, there are a number of challenges associated with anti-idiotypic antibody generation. To generate successful anti-idiotype monoclonal antibodies, appropriate immunization and hybridoma screening strategies are critical. For the generation of anti-idiotype polyclonal antibodies, the key factors are immunization and affinity purification. In terms of immunization, in some cases, the CDR region of the antibody may not be very immunogenic, meaning the antibody antigen may be unable to elicit an adequate immune response. This response is dictated by whether or not the immune system can recognize the antigen. Another obstacle to the generation of anti-idiotypic antibodies is the fact that the percentage of anti-idiotype antibodies in hyperimmune antiserum is very low. This can be compounded by a situation in which most of the antibodies raised target the Fc region of the antibody antigen. During the hybridoma screening process, an anti-idiotype clone, which coexist with a non-anti-idiotype clone, will be missed if conventional screening is applied. So, how do we overcome these obstacles to successfully generate anti-idiotypic antibodies for critical drug development studies? One of the first solutions to address the lack of immunogenicity of the CDR region is to use an antibody-drug KLH conjugate or Fab fragment as immunogen. The advantage to using Fab fragments is that constant heavy chain determinants, which can induce unwanted antibody responses in immunized animals, are absent. One must be sure, however, to confirm that your Fab fragment retains its antibody activity. At GenScript, our scientists typically use full antibody for monoclonal anti-idiotype antibody production and Fab fragments for polyclonal antibody production. Whether or not you choose monoclonal and polyclonal anti-idiotypic antibodies depends on your application. For pharmacokinetic studies, often monoclonal antibodies are preferred. Another method to circumvent the CDR region lacking immunogenicity is to adjust the immunization schedule or consider a different animal host. GenScript has a broad range of animal hosts available to ensure optimal anti-idiotypic antibody production conditions. As stated previously, during the hybridoma screening process, an anti-idiotype clone which coexists with a non-anti-idiotype one will be missed if conventional screening methods are applied. To overcome the issues surrounding the low percentage of anti-idiotypic antibodies in serum, it is critical to use a proper secondary antibody, such as anti-mouse Fc, to prevent cross-reaction with human immunoglobulin. In addition, high-throughput binding screening and capture ELISA assays can be used to select the natural epitope recognized by the antibody. In a capture ELISA, the target antibody antigen is lifted in solution instead of directly coating on the plate in order to avoid epitope denature or masking. Selecting the optimal anti-idiotypic clone or clones from numerous anti-idiotypic antibodies is perhaps the most critical step for successful anti-idiotypic antibody development. Methods for detecting and monitoring anti-drug antibodies demand reagents and controls with high specificity and sensitivity, particularly when the drug is a monoclonal antibody. In fact, often a range of affinities is desired for certain types of assays. At GenScript, a customer can choose the type of anti-idiotypic antibody they would like based on their application. One of the first methods used to facilitate this process is antibody pairing. In this case, one anti-idiotype antibody is used to capture the drug and the other anti-idiotypic antibody is used to detect it. We will see an illustration of this in the next few slides. Next, affinity ranking may be useful in situations where a broad range of affinities is desired. For pharmacokinetic studies, high affinity anti-idiotypic antibodies can increase assay sensitivity. However, for immune response studies, validating the assay with anti-idiotypic antibodies of different affinities can prove the assay is able to detect anti-drug antibodies in different affinity ranges. In epitope binding and antigen blocking assays, which is illustrated above, anti-idiotypic antibodies compete with antigen for drug binding. In pharmacokinetic studies, if customers have both antigen blocking and non-blocking anti-idiotypic antibodies, they can quantitate the serum drug level both in antigen-free and antigen-bound states. Determining whether the target antigen is saturated by the antibody drug is vital in drug monitoring assays. To expand on epitope binding for optimal clone selection, I have included an illustration and examples of the binding process. Epitope binding is a competitive immunoassay used to characterize and sort monoclonal antibodies against a target protein. Antibodies against a similar target are tested against all other antibodies in the library in a pairwise fashion to ultimately obtain a competitive blocking profile for each antibody of interest. Closely related binding profiles indicate the antibodies have the same or closely related epitopes. Thus, these antibodies are bind together. At GenScript, our anti-idiotypic antibody development protocol builds off of our proven success and experience with traditional monoclonal and polyclonal antibody production methods. Appropriate immunization, purification, and hybridoma screening strategies are critical for successful anti-idiotypic polyclonal and monoclonal antibody development. At GenScript, our scientists utilize our proprietary technologies to ensure the high specificity and affinity of anti-idiotypic antibody, including special adjuvant and immune mediators, supersensitive hybridoma screening, and optimal clone selection processes. We've discussed the obstacles and solutions related to anti-idiotype antibody generation. Now let's put everything into practice and delve into downstream assay development utilizing these powerful antibody tools. Due to their different binding modes and levels of affinity, anti-idiotype antibodies offer flexibility for specialized assay development. Here I will reiterate the three main types of anti-idiotypic antibodies that can be useful in a variety of capacities in drug development assays. Having both antigen-blocking and non-blocking anti-idiotypic antibodies allows the quantitation of antibody drugs in circulation, while complex specific anti-idiotypic antibodies, illustrated on the far right, can offer insight into the amount of bound antibody drugs in serum. This table is a useful guide for how to use concentration data obtained using a combination of anti-idiotypic antibodies. In the preclinical development stage, therapeutic antibody concentrations are used to understand pharmacokinetic behavior in non-clinical samples and to project the exposure of the first in-human starting dose. Ligand concentrations are used to understand the dynamic relationship of therapeutic antibody to ligand in order to assist in the identification of the biologically active therapeutic antibody concentration. Depending on the information required for certain decision-making regarding drug development, the data required and hence the selection of assay may differ at each stage of drug development. Both pharmacokinetic and immune response assays are useful bioanalytical strategies to generate data to address key concerns during the drug development process. First, we will cover the use of anti-idiotypic antibodies in pharmacokinetic or PK assays. The main purpose of PK assays is to detect and quantitate human antibody drugs in serum. IgG is the most abundant immunoglobulin in human serum, making up approximately 75%. Of that class, IgG1 makes up approximately 66%. The total serum concentration of the IgG1 subclass is between 5 to 9.5 milligrams per mil. The required sensitivity to detect antibody drug is in the nanogram per mil range, meaning assays must have the ability to accurately detect antibody drug despite a millionfold excess of similar molecules. Agents with this degree of specificity include antigens, or the drug target, and anti-idiotypic antibodies. The study of the tissue distribution and PK of antibody drugs requires the development of methodologies to differentiate the administered antibody drugs from endogenous antibodies. The anti-idiotype capture sandwich ELISA is one of the most popular assay formats. In this assay, ELISA wells are first coated with anti-idiotype antibody as the capture, then incubated with antibody drug in serum. Detection is carried out using labeled anti-human IgG. If one has both antigen-competing and non-competing anti-idiotypic antibodies, one may quantitate the levels of antibody drugs in the circulation, both antigen-free and bound forms. A more conventional approach for PK studies of antibody drugs includes the antigen capture ELISA depicted in Figure 1. The disadvantage of this format is the coded antigens may alter the conformation to some extent, thus affecting binding affinities and the resultant assay sensitivity. In Figure 2, the anti-idiotype bridging assay is an excellent example of using the same or two different anti-idiotype antibodies to detect and measure antibody drug. For this type of assay, a highly specific anti-idiotype antibody is immobilized at low concentration and captures the drug in the serum sample. The bound drug is then quantified with a second labeled anti-idiotype antibody that binds to the free idiotope of the drug. In a slightly altered version of this assay, one can detect bound drug using labeled antigen, illustrated in Figure 4, the anti-idiotype antigen bridging assay. The next type of downstream utilization of anti-idiotype antibodies is immune response or IR assays. Therapeutic antibodies are frequently immunogenic in animals. Immunogenicity in animals is not predictive in humans. However, information regarding immunogenicity in animals may be useful to interpret non-clinical toxicology and pharmacology data. An important part of the consideration of any biological therapeutic approach is evaluation of immunogenicity in anti-drug antibodies data. Due to the size of some clinical trials and the importance of testing patient samples at several time points, the FDA recommends a multidisciplinary approach. A rapid screening assay should be used initially, and any samples testing positive should then undergo a confirmatory assay such as ligand or antigen competition assays. It is up to review experts to evaluate the sensitivity and specificity of the assay itself and the characterization of the immune response, for example, whether anti-drug antibodies neutralize the activity of the product. Clinical reviewers must consider whether the immune response alters the serum levels of the product, affects clinical safety and efficacy, or both. Several aspects of IR assay development include high sensitivity and the ability to detect all isotypes. Other considerations include the avidity of the control used to evaluate the assay, as well as the effects of the matrix used to perform the assay. For immunogenicity screening assay development, the recommended assay format is the bridging ELISA. In this type of assay, antibody-drug is coded and detection of anti-drug antibodies is done with labeled antibody-drug. One important thing to note in this type of assay is the importance of the ratio of capture to detection of antibody-drug. For all IR assays, one must keep in mind that positive controls are necessary for proper assay development. If non-primate animals are immunized with a full-length antibody drug to develop a positive control, the antibody response will likely be against the FC region. Such a positive control may not be relevant for the anticipated immune response in human patients, where the response to antibody drug is primarily to the variable regions. Ideally, the anti-idiotype monoclonal or polyclonal antibody is the best positive control for this type of anti-drug antibody study. In addition, 3 different anti-idiotypic antibodies with varying affinities are a useful approach to mimic the natural immune response, wherein the antibody with the best affinity has the highest sensitivity. In addition to bridging ELISA assays, a number of other IR assay formats may be used, each having distinct advantages and disadvantages. Bridging ELISAs and radioimmunoprecipitation assays are sensitive, inexpensive, and typically require basic laboratory equipment. However, they may not be able to detect early immune responses and can be influenced by high levels of circulating drug. Conversely, surface plasmon resonance is the preferred method for detecting early immune responses and has antibody characterization capabilities. Its utility, however, is hampered due to the expensive equipment required and low sensitivity compared to other formats. Finally, electrochemiluminescence assays have the advantage of sensitivity but may lack the ability to detect rapidly dissociating antibodies. Thus, assay choice depends largely on the stage of drug development and access to necessary equipment. GenScript makes this process easy, however. Our anti-idiotype antibody development services offer readily integrated downstream antibody-drug, PK, and IR assay development. I would now like to take a few minutes to share a few successful case studies of GenScript's anti-idiotype development services. In the first case study, 10 anti-idiotypic specific monoclonal antibodies were obtained. ELISA data, in which antibody drug was coded on the plate to detect anti-idiotypic antibody, shows that these clones specifically recognize target antibody, but not negative screening material. In the second case study depicted here, anti-idiotypic high-affinity monoclonal antibodies were obtained. The affinity of anti-idiotypic antibodies were measured by Biocore T200 with the format of capture being the anti-idiotype antibody on the sensor chip and the antibody drug allowed to run through the sensor. The result of quick K-on and slow K-off rate indicates the high affinity of these anti-idiotypic antibodies. Finally, the last case study illustrates an antigen blocking assay where the anti-idiotype antibodies were grouped into antigen blocking and non-blocking binding modes. Our antibody experts here at GenScript have extensive experience developing high-affinity, high-specificity anti-idiotype antibodies for use in PK and IR assay formats. Additional features of our anti-idiotype antibody platform include high-speed production with 2 to 3 months turnaround time, antigen ligand blocking, epitope binning, and antibody pairing assay capabilities, both anti-idiotype monoclonal and polyclonal antibody packages available, PK and IR assay development, and a proven track record of 100% success for anti-idiotype antibody development. GenScript offers the development of both polyclonal and monoclonal anti-idiotypic antibodies. Polyclonal anti-idiotype antibodies from goats or rabbits are suitable when a lower amount of antibodies are needed within a short time, such as for pre-studies. If the anti-idiotype antibodies will be used in long-term studies under validated conditions, then monoclonal anti-idiotype antibodies are recommended. To get started, simply supply us with your target antibody drug and we will deliver your hybridoma cell lines and supernatants or purified anti-idiotype antibody within 2 to 3 months. If you have any questions or would like to have a discussion on your custom anti-idiotype antibody project and downstream assay development, please contact us and our PhD level technical account manager can help you. You may wonder why you should choose our services. Well, I'm going to go over our major advantages that made GenScript stand out from our competitors. The primary advantage of our antibody services is that we offer our customers a comprehensive one-stop service for antibody development. All that is required from you is the target protein sequence, and we use our years of expertise to design, optimize, and produce either protein or peptide antigen, optimize antibody development, and deliver you the purified antibody. To optimize your antibody production, we use our proprietary Optimum Antigen Design tool, which takes into consideration sequence length, hydrophilicity, surface orientation, flexibility, continuous epitopes, secondary structures, and sequence motifs, as well as coupling strategy. This Optimum Antigen Design tool integrates other available commercial software packages with our own algorithm to produce the most specific and highest affinity antibodies possible. One concern you may have for custom antibody production is that the antibody will not work or the yield might be low. At GenScript, the success rate of our custom antibody development is so high that we offer guaranteed packages. We guarantee the amount of antibody or hybridoma, ELISA titer, and Western blot results depending on the chosen package, which means that if we are not able to deliver antibody with the guaranteed packages, there will be no charge to you. The process is virtually risk-free. Another big advantage with our antibody services is our turnaround time, which is the fastest in the industry in terms of delivering purified polyclonal antibodies or developing specific hybridoma for monoclonal antibody production. You can get your purified polyclonal antibody or hybridoma in just 45 days, and all of our antibodies are produced in our ALAC accredited and OLA certified facilities. These are the things that make us stand out from our competitors. To give you a peek at our most popular antibody services, I included the following 2 slides. You can also find such information on our website. In addition to anti-idiotype antibody development, we offer a full spectrum of antibody services, including express 45-day polyclonal and monoclonal antibody production packages, standard polyclonal services in rabbits, mice, rats, chickens, or goats, premium hybridoma services, phosphospecific antibody services, large-scale antibody services, and a variety of specialized services. So far, there are over 500 peer-reviewed publications citing our antibody services. Many of these are in top-tiered journals. Here are some recent publications in PNAS, Cell, and Nature in which the antibodies developed by GenScript were used to generate the data in the paper. Besides custom antibody services, we offer a broad range of services to make your research easy. This slide gives you a general overview of the services we offer at GenScript, including gene synthesis and subcloning, peptide synthesis, protein expression and purification, custom antibody production, discovery biology services, and stable cell lines, all with the goal to make research easy. More than 9,000 peer-reviewed journal articles have cited GenScript services and products, making GenScript the most frequently cited biology CRO in the world. Now at this time, I would like to thank you for attending this webinar. I hope you found the information useful. Please complete the survey that will pop up on this screen when this webinar ends. If you have any questions or comments on what we've talked about today, you can email me at michelle.parker@genscript.com. This webinar will be archived on the GenScript website. Other webinars are also available there for on-demand viewing, or if you would like to simply download the slides for viewing, you can do that from there as well. Whenever you are ready to start your custom antibody development project, you can go to our website and use the online quote form, or if you prefer, you may email us at antibody.genscript.com. Thanks again. I am now going to check the chat window to see if there are any questions. Again, if you prefer, you may email me your questions and I will get back to you as soon as possible. Okay, we have a few questions that have come in. I will take a few of them. Please keep in mind our technical specialists are always available 24 hours a day should you have any additional questions in the future. Okay, our first question. How likely is it that an anti-idiotype antibody carries an internal image of the original antigen, such as whether or not it can mimic the eliciting antigen? Well, antibodies that carry an internal image are probably only a small fraction of all anti-idiotype antibodies. Once they are produced and characterized, they are exceptional experimental tools. While some anti-idiotype antibodies bind to the same receptors as the antigens, that by itself does not guarantee or provide proof that they carry a detailed image of the antigen. Given the variety, the next question is talking about the variety of anti-idiotype antibodies that can be used in downstream assay development and whether or not our anti-idiotype antibody packages actually allow a customer to choose the type of anti-idiotypic antibody. Yes, actually, a customer can choose the type of anti-idiotypic antibody that they would like to generate based on their application. For example, if a customer needs antigen blocking or different degrees of affinity for their anti-idiotypic antibody project, our GenScript project managers can guide them in ensuring that they select the appropriate anti-idiotype antibody for their assay needs. Our next question is whether or not it's possible that different species may give very different results in terms of the antibody-drug target antigen ratio that's determined in PK studies. Yes, actually both species and disease status can affect the target antigen profile. Even in the presence of highly characterized reagents, it should be understood that the antibody-drug target antigen exists in vivo in a dynamic equilibrium. Thus, ex vivo assay conditions can impact both antibody-drug and target antigen concentrations being measured, and also the equilibrium of the two. Thus, experimental results should really be evaluated to understand if they truly reflect the binding relationships in the study samples. Okay, so the next question is regarding whether or not LC-MS methods could be used to quantify antibody-drug. Yes, there are some publications which use LC-MS. The method, though, involves enzyme digestion to reduce monoclonal antibody drug into small peptides in order to be within the range of the mass spectrometry. However, the antibody must be denatured before enzyme digestion, which results in dissociation of the antibody from the target antigen, of course. Thus, using this method would only allow you to quantify total antibody drug in circulation. And I'll take one more question here. So it's regarding how difficult it is to generate other types of anti-idiotypic antibodies, such as the non-blocking versus the competitive anti-idiotypic antibodies. So certainly it is more challenging to develop these types with what we would consider somewhat rare specificity. Part of the reason is that they are more difficult due to the fact that fewer epitopes are actually available. Typically there will be many standard or antigen blocking anti-idiotypic clones to choose from, with fewer clones representing the other binding modes. Yes, and we have a question about whether or not these presentation slides will be available. Yes, they will be uploaded to our site where all of our webinars are listed on our GenScript site. So yes, definitely. Okay, well, I'd like to thank you all for attending, and we will be ending the webinar now. Thank you very much.